Protein formation, or protein synthesis, is a fundamental biological process that occurs in all living cells. It involves two main stages: transcription and translation. Here's a detailed look at the mechanism of protein formation. Transcription is the process by which the information in a segment of DNA is copied into messenger RNA, mRNA. This occurs in the nucleus of eukaryotic cells and in the cytoplasm of prokaryotic cells. Firstly, during initiation, the enzyme RNA polymerase binds to a specific region of DNA called the promoter, causing the DNA strands to unwind and separate. Following this, RNA polymerase, along with other transcription factors, forms a complex that initiates the synthesis of RNA. In the elongation phase, RNA polymerase moves along the DNA template strand, adding complementary RNA nucleotides A, U, C, G to the growing mRNA strand. The mRNA strand is synthesized in the 5' to 3' direction. Finally, in termination, when RNA polymerase reaches a terminator sequence on the DNA, it releases the newly formed mRNA strand. In eukaryotes, the primary mRNA transcript undergoes several modifications. A modified guanine nucleotide is added to the 5' end in a process called 5' capping. A tail of adenine nucleotides, known as the poly-A tail, is added to the 3' end. Lastly, introns, or non-coding regions, are removed, and exons, or coding regions, are joined together to form the mature mRNA. Translation is the process by which the mRNA is decoded to produce a specific polypeptide, or protein. This occurs in the cytoplasm and involves ribosomes, transfer RNA, tRNA, and various other factors. During initiation, the small ribosomal subunit binds to the mRNA near the start codon, AUG. The initiator tRNA, carrying the amino acid methionine, binds to the start codon. Subsequently, the large ribosomal subunit joins the complex, forming the complete ribosome. In the elongation phase, the ribosome reads the next codon on the mRNA, and the corresponding tRNA, carrying the appropriate amino acid, binds to the codon. The ribosome catalyzes the formation of a peptide bond between the amino acid on the tRNA in the P site and the amino acid on the tRNA in the A site. The ribosome then moves one codon down the mRNA, shifting the positions of the tRNAs within the ribosome. The empty tRNA exits from the E site, and the tRNA with the growing polypeptide chain moves to the P site, making the A site available for the next tRNA. Finally, in termination, when the ribosome reaches a stop codon, UAA, UAG, UGA on the mRNA, release factors bind to the ribosome. The completed polypeptide chain is released from the tRNA, and the ribosomal subunits dissociate from the mRNA. Here is a simplified labeled diagram to illustrate the key stages of protein synthesis. Transcription, DNA. Summary of key points. 1. Transcription initiation: RNA polymerase binds to DNA and unwinds it. Elongation: mRNA strand is synthesized. Termination: mRNA is released and processed. 2. Translation initiation: ribosome assembles on mRNA. Elongation: polypeptide chain grows as amino acids are added. Termination: polypeptide is released when stop codon is reached.